my name is Ashlyn Murray. I'm talking about a journal comparing the gut microbiome of 3 Anura frogs from a mountain stream in China. The study investigated these 3 frogs, here shown on the left. In descending order, it's 1, 2, and 3. So from this, they were trying to find if gut microbiome transmission was able to be quantified or if they were able to see it. And the benefits of gut microbiome transmission is it allows other species to broaden their dietary niches. This means that they are not set on one set specific diet, and they're able to branch out more with other foods. The scientists started by trying to find quantitative ways to find each frog's phylum, genome, and species, and they did this through DNA extraction and 2-step 16S rRNA sequencing. The results are found on the graphs on the left. In descending order, it goes phylum, genome, and species. So as you can see on the very top graph, it's much broader. There's very few variations, and as you descend down, it gets a lot varied and a lot more varied. Excuse me, and much more specific. They were able to find that the largest percentage and the largest quantity of phylum or bacteria in a specific phylum was the protobacteria, which is present in large quantities for all 3 frogs, if you see at the very top graph. And if you see in the bottom graph, on the bottom right, that was the rRNA sequencing. Now, when they did the relative abundance of gene functions, they found that the most enriched gene function was related to metabolism. So it's from this that they assumed, or they correlated, that the large amount of protobacteria was in relation to metabolism. Now, from this data, they were able to find correlations between the variety from frogs 1 and 2 and 2 and 3. This is because frog 2 showed a lot more variance in its gut microbe. So from that, they were able to determine that frog 2 most likely had a lot of gut transmission or microbial transmission from other species like these 2. And that's what's led to such a variety in their gut microbial environment. At the end of the paper, they did state that they think this is good evidence, but they don't believe it's enough evidence, and they attribute that to time sampling and would say that another experiment introducing that factor would be beneficial in determining gut microbial transmission. But it was a really entertaining journal. I had a lot of interest figuring out what these graphs meant and how they correlated, but it was really interesting overall. So, thank you.